All right. Hello, everyone. Hello, guys. Hope you're enjoying your vacation, your little summer vacation here. I'm just going to drop this little summer work for you guys to do so we can be on par with the curriculum, right, with the course outline. It's not too much that I'm going to talk about. It's basically the whole concept of enzymes, their characteristics, and factors that influence enzymes. And then when you guys come back, we'll talk about energetics in terms of chemical reactions, chemical energetics in terms of photosynthesis and cell respiration. Again, not as detailed as we did in the pre-WACE phase, right? With the electron transport chain, if some of you remember. So yeah, I just want to go over enzymes for this kind of summer homework now. Okay. So again, everything is in the OneNote. All the notes that you have, it's just a matter of taking a look at the video, understanding what I'm talking about. When we come back, I will not go over this content, but you will have it as a video. But if there's anything that you don't understand, we can spend a day talking about this topic, right, and answering any questions that you guys have, right? So if you remember, we were talking about the biomolecules and how different types of organisms can create different types of molecules, right? And there's different groups of them that are essential to living organisms, which are protein, carbs, lipids, and nucleic acids, right? So I went briefly in here, right, talking about the two types of nucleic acids, which is basically DNA for deoxyribonucleic acid and RNA for ribonucleic acid. And the difference between them is basically the sugar, which in this case for RNA is something called a ribose, which is a 5-carbon sugar. And for DNA, it's called deoxyribonucleic acid, right? In Unit 3 of the WACE program, we get into DNA in incredible detail, right? So you don't need to know the specifics for now. And so nucleic acids, they're usually a single strand of DNA, but of nucleic acid strand, right? And but DNA is special that it's a double-stranded molecule, right? And nucleic acids in general, they have their own building blocks, which are nucleotides, right? And these nucleotides are made up of little components that are referred to as the phosphate group, the nitrogenous base, and the sugars, right? Now, you don't need to know that for now, but just know that nucleic acids, they are made up of building blocks called nucleotides, all right? Nucleotides. And there's different types of nucleotides based on the nitrogenous base, right? And there's 4 different types, basically the letters that you see here, A, C, G, and T. And these nucleotides, these Legos, can be in repeating order. You can have several sequences or orders of these nucleotides, like AAA, TTC, GCAT, right? In any order of repeating nucleotides. Ultimately, for now, I just want you to know that nucleotides make up DNA, right? And also RNA, with a little difference there between the T and another nucleotide with a U. Ultimately, what's important is that nucleotides and nucleic acids contain information to create proteins, right? And when you're talking about the molecular level, that's referred to as coding, right? So a particular sequence of nucleotide codes for a particular protein, right? And we're going to see that. It's pretty interesting how that works, and so forth. So, and one last thing is that DNA, being so important that it codes for protein and pretty large, it's encapsulated by the nucleus, right, and the nuclear membrane to keep safe, essentially, right? So whenever the cell wants to make new proteins, it relies on an intermediate form to relay information, which is RNA. More specifically, something called messenger RNA, right? Which goes ultimately to ribosomes to help with the manufacturing, right? Or to the ribosomes for manufacturing to create the actual protein, right? And the last key point of nucleic acids is that the order of DNA determines the order of RNA, which will determine the order of proteins. Okay, so for now, that's all you need to know. We'll go into it in a lot more detail. Before we get into that kind of stuff, we need to understand a little bit broader level basics, right? And that, let me just go up here. All right. So the basics meanings like biochemical pathways or processes in general for the cell, right? So living organisms, single-celled and multi-celled organisms, are essentially based on chemical reactions. And all the chemical reactions that are occurring in your body is referred to as metabolism, right? And these chemical reactions could go in a series where reaction 1 basically will produce or create products for reaction number 2, and then make a product for reaction number 3, and so forth and so forth. And ultimately, you have a series or a chains of chemical reactions, right? But ultimately, everything that's being produced by these chemical reactions, one way or another, are needed by the cell or needed to be removed by the cell. Okay, so here's a little depiction of what happens, right? Let's say you eat some food, right, and you get the materials broken down from the food, and one way or another, you have some particular pathway that you can use that to either create food, or you can use it to break down other stuff, right? And these two types of reactions are further broken down into what are called anabolic reactions and catabolic reactions, right? And anabolic reactions is basically the process where you have the synthesis, right, or the creation of complex molecules from simpler ones, right? So let's say we broke down all our food into simple sugars, simple amino acids, and then we use these little building blocks to create more complex molecules, as you see here, right? E, D, and F. And that's referred to as an anabolic reaction, and moreover, anabolic pathway, right? At the opposite side, you have catabolic reactions, where you're basically getting complex molecules and you're breaking them down into simpler ones, right? That's called catabolic reactions, or catabolism. Okay, and for anabolic reactions, you could also refer to as anabolism, right? And so one way or another, these two types of reactions are going to occur in the body. It's all a matter of physical molecules, either you're creating more complex ones or simpler ones. Okay, and then ultimately, for that to happen, it generally occurs in a series of reactions. You seldomly, if I can say, will just have one simple reaction, and then that's the final product. In biochemical situations, right, in the cell, you're generally going to have that in a series, right? And that's referred to as a biochemical pathway, right? Now, different types of molecules require different biochemical pathways. There's many millions of biochemical pathways in the body, right? Some are more vital to the organism's homeostasis than others, right? But nevertheless, you have many, 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 right? To give you a sense of what I was talking about, you have this little depiction here where you have the initial reactants, right? When they react together, they make product A. And then one of the things that we're going to talk about is that in order for a series to continue on, where you have one reaction to another, they're generally aided by molecules that are called enzymes, right? And these enzymes are very special in the sense that they regulate, sorry that I have this here, that they regulate how fast the reaction can occur, right? And they're very, very specific. is you see here depicted in color, right? This enzyme in orangey, pale orange here will only work for products A that basically emerge from the initial reactants, right? And nothing else. So these enzymes help in the reaction between these products to produce these products, right? And then another enzyme, enzyme B, or this greenish enzyme, will come in and work with all the products B, maybe along with some other molecules that we're going to talk about, to produce product C, and so forth and so forth and so forth, until you get product D, right? In essence, this is a biochemical pathway, right? They're regulated by enzymes, okay? And it's very important to have enzymes because they essentially facilitate the reaction to progress relatively quickly. You need things to happen pretty fast, right? Because if things happen slow, you could have some derailment of kind of equilibrium in the body, in the cells, right? If that happens, then the whole cell will stop functioning correctly, and it could even, destroy itself. So, yeah, they're quite essential, right? So, some examples of where you would see this type of pathway in the use of enzymes, right, is whenever you collect a lot of glucose. In the case of plant cells, you get glucose, and also for animals. And glucose, if you have too much, and that simple sugar, right? It could be in the blood or in the vessel system of the plants, and it can get too thick. It can make the solution too thick. So you want to have it in a very insoluble form that you can store it somewhere, right? In this case, glucose can be converted into a stored form that we know as starch, right? And that way you have like a supply of it, a continuous supply of it, and it's not harmful to the plant itself. Okay, and that way the plant can keep producing and using whenever it needs, right? Ultimately, when you need to use glucose, you basically undergo a special process called cell respiration to extract as much energy from glucose, right, to produce ATP. Now I'm getting a little bit ahead of myself, but yeah, a byproduct of this burning, if I can say, of glucose in cell respiration is carbon dioxide, which ultimately is liberated into the atmosphere and used by plants, right, via photosynthesis. Again, all this we're going to see in much more detail, so don't worry if you don't understand the context yet. Okay. Nevertheless, what I want you to understand is that these chemical processes, right, these biochemical pathways, they're tightly regulated by enzymes, right? And that's one of the things we're going to talk about in the next section here, right? Also, not just enzymes, but if you remember when we talked about in the pre-WACE phase, being eukaryotic cells, there are many organelles that have membranes, right? So they're compartments, right? They're little microenvironments, and these microenvironments enable the cell to become more efficient, because if you have, let's say, the mitochondria working on something, you can have the ER working on something else. So if you have the mitochondria and the ER, and let's say they want to produce different things and they need different chemicals, different chemical reactions are occurring, by having organelles and by having compartments, this enables the cell to be more efficient. One organelle is working on something else while the other one is working on another thing, right? So by creating these little organelles and having these little microenvironments, it enables the cell to be more efficient, right? Especially when it comes to recycling things like lysosomes, right? That they have digestive enzymes within these little vesicles, specialized vesicles, and if these digestive enzymes come out, the cell can even kill itself, right? Which does happen. Okay, it also helps reactions to occur at the same time, right? Different products occurring, again, overall for enhanced efficiency, right? Not just that, but the products that are occurring in one organelle could be stored for later use, or you can have a buildup of concentrated products that you can use later, right? Basically concentration gradients, okay? And that we're going to take a look at also in a little bit. Yeah, so an example is like the mitochondria, aside from just being a compartment, one of the things you're going to see many, many times that I've started to mention before is surface area. By having many folds and stacks like the ER, that increases and enhances the rate of chemical reactions occurring, right? So this is just a little intro of what I want to talk about for this video. Okay, let me go to the next one here. 10.2 basically is on enzymes. Okay. So, oops. All right. Just move that a little bit here. So, what are enzymes, right? So these are molecules. They're generally proteins, right? And they essentially work by lowering the activation energy. If you remember the activation energy there, that I'm going to talk about also in a bit. But essentially, they regulate biochemical pathways. Okay, they're referred to as biological catalysts because there are some inorganic catalysts that you've probably seen in chemistry, right? And the term enzyme, you really don't need to memorize this, but just a little bit of more knowledge. Edward Buchner, right, is the scientist that coined the term enzymes, right, after working with yeast cells to basically study glucose fermentation, right? And essentially, like I mentioned before, enzymes are organic catalysts, and the whole function of that is basically to speed up reactions, chemical reactions. In this case, biological reactions. And one characteristic about that is that enzymes don't really react with the other reactants, right? They just help. Let's say if we have reactant A and B, these enzymes, they just help them kind of merge together or break apart. But the enzyme itself doesn't become part of the product, right? It just helps out, okay? So that's what I mean by without undergoing any change itself, okay? And most enzymes are proteins, are different types, but they're called differently. They're like ribozymes, but that's a different thing, right? Mostly they're proteins. And all enzymes are macromolecules, so they're generally big, right? And the whole function of it is to lower the activation energy that we're going to talk about in a bit, right? A little bit of just kind of reminder there. Maybe some of you know this little symbol in mathematics. This is the sum. The sum of all chemical reactions is known as cellular metabolism or metabolism, right? And of course, you're having different chemical reactions, different types of cellular reactions occurring at different times, at different rates, right? But yeah, you do have a variation of chemical reactions. And it doesn't happen by chance, right? Everything is tightly regulated, okay? Depending how the reactions, how fast they go, it will enable the cell to do something particular, right? To break down food, to produce enzymes, to break down food, to produce ATP, and things like that, right? To divide, for the cell to divide, to undergo protein synthesis. So everything every reaction occurs in its particular time at a particular speed, right? And that, of course, again, regulated by enzymes. Okay, so why are enzymes so important? Essentially, without enzymes in the body, the reactions will be way too slow, right? The chemical reactions inside an organism would be incredibly slow that the organism wouldn't even be able to live, right? And another characteristic is that enzymes can also control the rate of reactions, right? Not just help it out, but whenever there's too much concentration of a particular substance A, let's say, these enzymes can come in and kind of like stop them from reacting, right? So they can regulate chemical reactions. And one thing about enzymes is that they're so special and they're so specific that there's generally one enzyme for any protein. So if we have like millions of proteins that regulate the body, we have enzymes that regulate those proteins, right? So they're very specific and they'll only work for one particular thing that we're going to take a look at. All right. So in terms of enzyme general characteristics, I put this little panel here that's in your book that basically talks about what it does. You try to memorize this. This is like the big pick of what you need to know for enzymes, right? And I want to go a little bit more detail in terms of catabolic and anabolic reactions. And in general, they're referred to as biochemical processes. The main metabolic pathways that we're going to focus on in terms of living systems is essentially photosynthesis and cell respiration, right? And each of them depict these corresponding reactions, catabolic and anabolic reactions, right? And ultimately, the whole point of these reactions is essentially to produce or transform energy into usable forms, ultimately in the form of ATP, right? So to give you a sense, photosynthesis, produces glucose, right, through light energy, light reactions, dark reactions occurring, yada, yada, yada. Ultimately, you produce glucose, but that glucose is then used to produce ATP via cell respiration. We're going to see this in a lot more detail, right? So in general, right, again, you have this little diagram here. You had food containing a mix of complex and simple organic compounds, let's say bread, right? If you remember these key concepts, digestion, absorption, assimilation, these molecules that come in, either they're going to go one route or they're going to go the other route. Either they're going to produce even more complex compounds or they're going to produce simpler compounds, right? Catabolic, anabolic, right? Synthesis, breakdown. And something that's related to this is endergonic reactions, exergonic reactions that I'm going to talk about in a bit. Okay. Again, one example of catabolic reactions is basically starch, where you have a bunch of sugar molecules that were linked together, basically making a polysaccharide, a starch. But when you want to use the energy from starch, you kind of snip it, right, from the tip and take out one glucose out of another, one at a time, right? That's a catabolic reaction. If you want to create proteins, big proteins, you basically get little simple amino acids. You start linking them together, okay? And until you create this macromolecule, all right? That's anabolic reactions. And ultimately, you can't really talk about this without having some form of discussion with the topic of energy, right? There is a relationship that when you create things, more complex things, energy tends to get absorbed. That is referred to as endergonic reactions. And there's also reactions that whenever you burn energy, chemical energy, or you break down molecules, you're going to release energy in the form of heat, right? This is called exergonic reaction. It releases energy, right? So in the case of photosynthesis and cell respiration, each of them does that particular thing. So cell respiration is an exergonic reaction, but produces ATP, and photosynthesis is an endergonic reaction. All that energy is accumulated and dumped into glucose to make it a very high energy rich molecule, right? And the book pretty well defines this whole concept that I've been talking about right now. A biochemical process or reaction occurs when the chemical bonds or inputs or reactions are broken and the atoms recombine to form new substances and substances, right? The output or products In endergonic reactions, a total net amount of energy is absorbed and locked up in the bonds of the products, which have more energy than the reactants. Okay, and I'm going to show you this in diagram form. In exergonic reactions, a total net amount of energy is released from the bonds of the reactants, and the products have less energy than the reactants, right? So one liberates energy, one absorbs energy. Okay, if we take a look at this graph here, some of you might remember this from S1, right? Ultimately, this concept of endergonic and exergonic reactions could be a little bit played with via activation energy, right? And okay, let me kind of move this here. So what is activation energy? I've kind of been mentioning this for a bit. Activation energy is essentially the amount of energy needed for reactions to actually react together, to transform and to create new molecules, right? There's a certain amount of energy you need in order to react, right? So in general, energy is the capacity to cause change, right? And it's measured in either joules or kilojoules or calories, right? And for any chemical reaction to start, right, it requires a certain amount of energy, which is that, right? The activation energy or energy of activation. So what is it? It's the minimum energy required to start a chemical reaction, okay? Usually the input of energy is heat, is fire, right? That's why we kind of use heat to produce or create a lot of things, right? So yeah, what it does actually though, it's that normally reactions, in the body, they require a relatively high amount of activation energy, right? So what enzymes do is that they help make the reaction go faster by lowering the amount of activation energy that is needed. Okay, so let's look at this graph here. Let's say you have these reactants, right? Reactant A and B, and they themselves have a certain amount of energy, right? But in order, and you put this, let's say you mix glucose and water, right? You have a sugary solution, and you want it to dissociate, to break apart, right? Glucose has a certain amount of energy inside its chemical bonds, but in order for glucose to dissociate, it needs to reach this amount of energy for the reaction to catabolize, if I can say, right? Okay, we see here on your right side, on the y-axis, is an amount of energy, progress of the reaction. Okay, so it would go pretty slow with this amount of energy. So if you put an enzyme in the system, in that solution, look how much the activation energy lowers, right? So the amount of energy that you would need for these two, for glucose to dissociate, to break apart, is essentially half of the original amount. If you take a look here. So what does that mean? It means that you need less energy for the reaction to forward, right? To continue, all right? If you need less energy, that means more glucose is being broken down in a certain amount of time. That's the definition of being faster, right? So that's what enzymes do, right? They hasten the reaction by weakening the chemical bonds so they can react better, quicker, okay? A specific example that we've done before, if you remember the liver experiment that we did, right? That's catalase, right? It's the enzyme that helps break down hydrogen peroxide. Basically this here, H2O2, hydrogen peroxide, will break down into water and oxygen, and it's helped by catalase, okay? Which, of course, it's incredibly important for cells to use because hydrogen peroxide can be a poison. So in this case, catalase can speed up a reaction 1,000 times with its original rate of reaction, okay? This panel that you see here is essentially the same thing I'm talking about, but without catalase, look how much energy you need for hydrogen peroxide to break apart, 71 kilojoules. But, right, if you add an enzyme, okay, if you add an enzyme, in this case catalase, you only need 8 kilojoules for the reaction to occur. Okay, so great difference, great difference there. Okay, so this is a visual depiction of how enzymes work, right? And before I continue with the depiction, I just want to clarify with that. Enzymes are not destroyed, they're not consumed by the reactants, they're not altered, or they could be temporarily changed a little bit, but ultimately they're not changed with a reaction, okay? And some things that you do need to remember is that enzymes, they work by having certain kind of areas where the reactant will come in and bind. All right, there's some terminology you need to know. So the first one's called active site. Okay, the active site, this little good, is where the reactant is going to come in and bind and join, right? Except that you don't really call them reactants. That's kind of weird when you're talking about enzymes. They're referred to as substrates, right? Substrates is basically any molecule that's going to come in and bind to the active site of the enzyme, all right? And it's very specific, incredibly specific. So if you see how this shape fits this little groove of the enzyme, when that happens, you create a temporary little complex that's referred to as the enzyme substrate complex. It's temporary, right? And then what happens is that the enzyme kind of provides a certain amount of tension in the chemical bonds between the itself and the substrate that it kind of weakens the bond that enables the products to form. In this case, you have the blue and the triangle shapes here that are liberated. So in this case, this is a catabolic reaction that got accelerated by enzymatic activity, right? So make sure you understand this panel. This is a very important one that you need to know. That's the general concept of enzymes, okay? I do recommend doing the questions again, right, guys? Remember, practicing this type of questions does help you review. Okay. This panel is quite important. Make sure you study this one, right? 10.3 is quite important. All right. So how the enzyme works is essentially based on the active site, the substrate, right? And that specificity between them, right? So before we get into that a little bit, I want to go over some kind of naming, nomenclature. Nothing serious, but just for you guys to know, anytime you see, maybe I have this a little bit too big. Anytime you see a molecule in the test or book or whatnot that ends in aces, okay, it's usually an enzyme, okay? The majority of enzymes end in the word aces, but not, it's not a rule, right? A 100% rule. There's some other different types of enzymes that end in, all right? Like trypsin, okay? So again, like I mentioned before, right? Enzymes are proteins and they have a very specific shape, meaning that they have a very specific substrate that they're going to attach to. All right. Very important for you to understand is that the shape of the enzyme defines the shape of the active site, will define which substrate will bind to it. Okay. You have your substrate. Once it binds, it'll create that complex. And the binding site is a very specific area, right? This is called the active site. All right. Some examples of these enzymes and that their end with aces are sucrase, lipase, maltase, nucleases, right? You're gonna learn some enzymes inevitably. Pepsin is an example of an enzyme that ends in IN, like I mentioned before. Okay, so the general gist of how enzymes work is essentially you have a substrate here. If we start on this side, okay, we notice this is a disaccharide. Okay, 6 carbon sugar, 6 carbon sugar, they're linked together, a disaccharide. This enzyme here comes in and will only catalyze this substrate, this enzyme, because of the binding site, that active site here. If you notice, look at the shape is just perfect for just this enzyme. It comes in, they bind together, and the tension that occurs between these two, okay, of the substrate is enough to weaken the bonds and break them apart. All right. The enzyme does change a little, okay, does change a little. This is called conformational change, where kind of it tensions the substrate to the point that they just break apart. And then the products leave, the enzyme comes in again, it's empty, so it can come in and get another substrate. So it can be actually reused, which is pretty cool, right? You don't need to waste energy to produce more and more enzymes, right? So the way that scientists think enzymes work is actually a little bit, there's two models, right? I don't want to say there's arguments or, tension between it, but there's just two proposed models on how enzymes likely work, right? You do need to know this also. The two models are the lock and key model and the induced fit model, okay? And the lock and key model is essentially this one that I discussed here, right? The shape fits perfectly with the substrate. The shape of the enzyme and the active site, moreover, fits perfectly with the substrate. It's referred to as the lock and key because it's a fixed groove. It's a fixed active site shape, meaning that it's incredibly specific and it will only bind to one particular substrate. Okay. You can see here, you have your little substrate molecule coming in, binding to the active site. You create this temporary substrate enzyme complex. And then just because of the tension, you have this product being produced, right? Or you can go in the reverse way. These two products, these two reactants come in, they temporarily bind to the enzyme, and the enzyme could provide energy for them to actually fuse together. Not all enzymes work in a catabolic way. They can also work in an anabolic way, okay? So that's the lock and key model. There's another model that's called the induced fit model, which is, it's more accepted. It's research and analysis have shown that this is the model that actually works better. It happens more, that happens more, right? It doesn't mean the other one doesn't happen, but it happens more. Okay. Now, what does this mean? What are some characteristics of this? Is that it has broader specificity, right? It undergoes bigger conformational changes, bigger changes physically to the enzyme temporarily. And the enzyme is lowered due to stresses on the substrate. A lot more stress is on the substrate. Okay. So what do I mean by this? All right. So in the induced fit model, you have that same molecule, right? Just for simplicity's sake to explain. And this model, this enzyme doesn't fit perfectly with the substrate, right? Or the substrate doesn't fit perfectly with the active site of the enzyme. If you notice in this case, if you see my little arrow here, the little curvature is almost perfect. But here, right, you have some little space here. All right, so the substrate comes and binds. Wait, what do I have here? Right, and the substrate comes in and binds to the active site. Sorry about that. Kind of like, give me a second, guys. For some reason, why do I have? Let me see. I think I have some effects on my face. Oh no, how do I get that down? I have a beard here. Let's see how much it changes. Almost nothing. Okay. Anyways, I thought I saw something flying around. That was weird. All right. Uh, anyways, so in the case of the induced fit model, induced fit, right, means that essentially you're creating the active site to mold itself toward the substrate. So they're not a perfect match, but the enzyme is a little bit flexible in that sense, in that it can mold itself like clay, if I can say, and mold itself and surround itself a little bit toward the substrate and then break it apart, right? So enzyme molecules change shape, closing around the substrate molecule, and then you make the products. All right. So because of that, let me write this down. I didn't write this down here. I'm gonna write slightly, right? Slightly different substrates can bond. So what does this mean? Slightly different substrates can actually come in and use the same enzyme. All right. If you have similar substrates that they're around the same shape, they can use the same enzyme. So this means that one enzyme can actually help different reactions, right? Different different substrates that are in the same class, if I can say, right? I'm going to explain that in a little bit as we go into more detail with molecules. All right, but just know that being able to induce the shape to fit, it enables more substrates to work with one enzyme. Okay. Lastly, here, this little side note here, enzymes can work in both directions depending on the substances. Yeah, enzymes could work in the reverse order where they can join, all right, and create one more complex substance from simpler ones. But it depends on a lot of factors, on concentration, on amount of energy, right? This is a little bit beyond the scope of high school, but just for you to know that enzymes could work in both directions. Okay. This is a picture that I put from another textbook. It's essentially the same thing, right? But you're taking a look at anabolic reactions, right? Anabolic reactions. So lock and key, there's no change in the enzymes, produces a more complex molecule. Induced fit model, slightly different, changes and creates that product there. All right. Good. And let's take a look here. All right. The last thing that I want to talk about for today is the factors that affect enzyme activity. There's not that many. There's 4 or 5. Yeah, and definitely you should understand that anytime you see factors, this is a key thing that you will or you can get assessed on in the WACE program, right? Factors enumerated. So remember that enzymes, they're basically they speed up the rate of the reaction. But having that type of role, there's definitely certain things that could influence how effective they could be, right? So since enzymes are very specific and they're in their shape, right? Changing their shape slightly can disable them from doing their job correctly. All right. So because of that, enzymes are very sensitive to environments, okay, and different environmental conditions. Okay, so some of these are temperature, pH, concentration, competition, right? All these are factors that can affect the efficiency of enzyme activity. All right, I'm going to go over them one by one. Okay, any of these factors can influence the enzyme's ability to function. The first one is temperature. All right, temperature. Remember, temperature is a factor that increases the rate of reaction, but if you heat up the enzyme too much, you can destroy it. All right, it's something called denaturation. Right, temperature can increase the reaction rate, but at some point it can denature the enzyme. Denaturing means to completely destroy the enzyme shape. All right. And but that doesn't mean that different enzymes have evolved to withstand different temperature conditions. Don't think that all of them work at a very nice, snuggly 25 degrees Celsius. No, different enzymes work in different environments. So here's some examples of some organisms that have enzymes that work in different temperatures. The first one is called psychrophiles, right? And these are bacteria, right, that work in very low temperatures. Enzymes work best at that temperature. But then once you get it out of this temperature range, it starts to decrease. The efficiency of the enzyme starts to decrease, right? The rate of reaction starts to decrease. Enzymes that work at our body temperature, basically at 37 degrees, 36.8 degrees, 37, all right, 37.5, they work best at our normal body heat. As you get them too cold or too hot, they will start denaturizing, right? So I'm going to talk about that in a bit. That's health related. And there's some enzymes that love to work under extremely hot conditions. Basically, let's say thermal vents, onsens, something like that. Incredibly high temperatures, they work best, right? These are called thermophile organisms, and they have very special enzymes to work in these conditions, right? And there's a special enzyme that we're going to use in the lab when we start doing the molecular biology sections that's called Taq polymerase, right? It's a special enzyme that actually is used to make copies of DNA, right? In a technique called PCR, polymerase chain reaction, right? And this enzyme works more at 70 degrees Celsius as opposed to 37 that we use, right? So all in all, I want you to know that enzymes can use different temperatures. Different enzymes use different temperatures, but when you have a stable environment like our body, the majority work under a very narrow condition, right? Or else they can denature. An example of denaturation is basically lobsters. When you catch a live lobster, right? They generally have this kind of metallic blue-purple look appearance, right? And what happens is that whenever they get thrown into the boiling water, right, to get cooked, these enzymes, they denature, they destroy some other pigments that create this kind of coloration. And then what's left is this very nice, yummy, delicious red coloration, which we all love to eat, right? That's what gives it its nice color. When you denature a protein, scientists have not fully discovered a way to bring it back to its original shape. So once you denature a protein, that's it, you kill it, you know? Protein is not reversible. Another example is basically eggs. When you open an egg, a raw egg, it's basically clear, right? The egg white, it's clear. But whenever you cook it, it turns white because you just destroyed the protein, right? To create it and one of the effects of destroying the protein is that the egg becomes white, and that's what we eat, right? That is denaturation. You denatured the protein. I believe it's albumin. Not 100% sure, but that is what is happening, right? I think maybe scientists have started to figure out a way to reverse renature. We can check that out, right? And you know, it's so important to regulate temperature and to regulate how enzymes work. They're especially important when you get an infection, when you when you get, let's say, a viral infection or bacterial infection and you get a fever, the reason why we get a fever is that we raise the temperature so the enzymes of the pathogen or the invader could get destroyed. But at the same time, we're kind of sacrificing our enzymes. So that's why they say don't get your temperature too high, or else it could be damaging for your body too, right? Yeah, raw meat also is, you can cure meat from decomposition by using enzymes, right? In the lobster example, that you really don't need to know. The actual chemical enzyme and pigment that's happening is it's astaxanthin, right? You don't need to know that. pH is another example. All right, so similar to temperature, if you get it out of their pH range, the enzyme could be denatured. All right, not necessarily, yeah, it could get denatured in this case because of the hydrogen bonds. The acids or bases can alter the chemical bonds that are interacting with the protein, right, to create the shape of the protein. So, even within our bodies, like the pepsin inside the stomach to break down the food that we eat can work in very acidic environments, right? 1.5 to 2 pH. But if you take it out of this stomach acidic environment, it won't work, right? But you have some other ones like the alkaline phosphatase in the bone. This works at a more basic pH this is related to bone structure and function, right? If you take it out of this high pH, it'll start to go down, the efficiency. And then you have others like amylase that you secrete on your tongue to start the digestive process. This works around a pH of 7, right? So again, similar to temperature, pH also works in that same way. If you take out the enzymes out of their environment, they're not likely going to work. They're likely not going to work. Okay. Aside from pH, there's a couple of more. Concentration. In this case, you're not destroying the enzyme, but you influence how fast the activity works just by simple concentration, right? If there's more substrate with the enzyme, more products are being made at the same amount of time. But there is a caveat there. If you have a fixed amount of enzyme concentration, and you can let's say you put fructase, which is the enzyme to break down fructose sugar, right? You can put as much fructose sugar, but if you only have like 10 enzymes, there's nothing much you can really do, right? So in terms of substrate and enzyme concentration, right, the reaction rate depends on how much enzyme you have, not substrate, okay? Even if you have excess substrate. Take a look at this figure here. True, as you add more substrate and you have that same amount of enzymes, the rate of reaction will occur higher, okay? But there's a point that all enzymes are being used. So even if you have an abundance of substrate, you just have to wait anyways because all the enzymes are being used in that particular time. When that occurs, that's referred to as a saturation point, okay? Saturation point. So that's concentration. Enzymes is another one that you probably have heard of in terms of neurotransmitters, maybe. In this case, you have a situation where you have what are called inhibitors. Inhibitors are substrates that could come in and compete, I want to say, not fight, compete for the active site for the substrate. So what I mean by this, let's say you have your enzyme and you have a substrate, okay, that wants to be broken down. So it could be, the products could be used for something else, for another reaction, right? But if you don't need this particular products at this particular time, then why would you bother to waste energy to break down this substrate, right? So inhibitors are used to basically stop the enzyme from functioning. It's like a plug, right? If you don't need it, turn it off, right? So these are called inhibitor molecules, and they come in and they bind to the enzyme until it's needed, right? When there's enough substrate that needs to be broken down and the products are needed again, then the inhibitor leaves. Okay, the way that it leaves is another issue, but this is what I want you to know, right? Inhibitors can come in and compete for the active site. Okay, the inhibitor molecule blocks the active site. You see this a lot in negative feedback mechanisms, right? And regulatory mechanisms in the body. For now, that's all you need to know. We'll see these mechanisms, regulatory mechanisms, in Unit 4. All right, so they compete for the active site. And the last one we're going to talk, and this will pretty much end the video, is cofactors and coenzymes. All right, these are opposite to inhibitors. So enzyme activity, right, can also be influenced by something that are called cofactors and coenzymes, right? If you notice, they have this word called collaboration. They're cooperating together, right? And this enhances the way that the enzymes works with the substrate, right? Sometimes enzymes are even present in an inactive form, and they can only be activated whenever these molecules are joined with them, right? There's two types of molecules, and they could be organic or inorganic. All right, I'm going to explain what that is. The first one is called cofactors, and they're essentially small inorganic molecules. ATP is one, zinc, iron are others, right? Basically minerals there. And they help facilitate the binding of substrates to the enzymes. Without these cofactors, enzymes don't work. This is pretty much supplements. So maybe you've seen your parents having some supplements, vitamins and minerals. They're basically taking cofactors and coenzymes, right? Coenzymes are larger organic substances that are generally carbon or lipid based, and they help bind to the enzymes to enhance a reaction, right? One way or another, you're helping the enzyme to be more active, okay? So yeah, many come from vitamins, and they help in many biochemical processes, okay? Some examples of like an example of deficiency, let's say if you lack vitamins and minerals, then you can start to get certain types of diseases. In this case, there's something called beriberi disease that you have a deficiency in thiamine or vitamin B1, which helps enzymes convert, right, to this particular molecule, thiamine pyrophosphate, which helps in the breakdown of glucose, right? So people that have a deficiency in this particular coenzyme, right, have a more difficult time breaking down glucose, right? So very important to have these cofactors. All right. So I hope I didn't take too long, guys. An hour should be enough. We want to progress as much as we can for photosynthesis and cell respiration that we're going to talk about. This will make us be right on time with our schedule. So yeah, hopefully it wasn't too exhausting. All right, guys, you don't need to answer any questions. Just make notes, come back to the class if you have any questions on it, because I'm going to continue straight into ATP and photosynthesis when we come back. All right, guys, thank you guys for watching. All right.